So our last speaker of the session is Yoni Haitin from the Faculty of Medicine, and he will be talking about molecular insights into KCNH1 channels, current inhibition.
Okay, last talk. Hang in there. Okay, hi everyone. I wish to thank the organizers. Okay, here we are. So I wish to thank the organizers for inviting me to give this talk. I'm a biologist. I will talk to you about biology and biological problems, so bear with me. So today I'm going to tell you a little bit about an ongoing project about dealing with the molecular insights into KCNH channel inhibition, and maybe expanding a little bit about the non-canonical roles of ion channels. So, but before that, I want to tell you a little bit about my new lab and what we're interested in. So we study ion channels, but we study them as signaling complexes. We want to understand how channels assemble with modulatory proteins and how those proteins help channels convey their important functions in cells. We are very interested in protein allostery. We use methods to allow us to follow the conformational wave associated with channel function in real time and in submolecular resolution. And finally, we use a variety of structural methods to elucidate ion channel structure at atomic resolution. We use X-ray crystallography, of course, and soon Cryo-EM. So how do we reach these goals? We study channels in all three angles of this triangle. We study ion channel function using electrophysiological measurements combined with fluorescence to really see the conformational changes that allow channels to conduct current. We use protein structure to see how these channels look, and hopefully in different conformations. And then we go up to the cellular level and see how channels affect cellular activity. So now to the background. We reach the background. What are ion channels? So ion channels are the engines of our electrical signaling in the body. They, of course, express in neurons where they conduct ions across the membrane, and this conduction and this separation of ions creates the membrane potential. This membrane potential leads to activation of some types of ion channels, the voltage-gated ion channels, and this activation or opening of the gates create the generation and propagation of action potential. This is the language neurons talk with. But that's not all ion channels do. Besides driving our electrical signaling in the body, ion channels over the past 10 years or so were discovered as a major contributor to cancerogenesis. You can see here the difference between normal cells and cancer cells, and one striking difference is the proliferation rate difference between these two cell types. And ion channels are indeed important for many aspects of this phenomena. They contribute to cell migration, adhesion, invasion, and cell cycle control. And this really fascinates us, how they do that. So the ion channel we are currently studying most, it belongs to a huge family of proteins termed the 6 transmembrane cation channels. This is a superfamily, and the family that we're interested in are called KV10-212. We have 3 subfamilies within this one. So the KV10-212 include 3 subfamilies, the KCNH, the CNG, and the HCN. All are crucial for numerous aspects of our physiology. For example, HCN are very, very important for normal brain function. Without HCN, neurons won't be able to integrate information, to sum information. CNG, or cyclic nucleotide-gated channels, are crucial for our senses of vision and smell. Without them, we won't be able to detect those stimuli. And finally, hERG are expressed in the heart, and they are crucial for normal cardiac function. Here you can see the cardiac action potential. This is the change in the membrane potential that leads to the contraction of the cardiac muscle. And as you can see here, the did I do that? Sorry. Okay. So IKR, the fast potassium current that is produced by hERG channels, which are part of the KCNH, terminates the cardiac action potential, and it must do so in a precise timing. Why is that? You can see here that when the termination of the cardiac action potential something is wrong with it, there is an elongation in the action potential. And this elongation is called long QT syndrome and can lead to very, very dangerous cardiac arrhythmia called ventricular fibrillation. This is a clinical death situation. So you can see here what happens to a soccer player having this mutation in hERG in the middle of the match. He just have this disastrous event and dies, basically, but he knew about that, and if you saw, he got a shock from a transplanted defibrillator, which saved his life. So he had a mutation in KCNH channel leading to death, basically. So what about the topology of these channels? How do they look in a very simplified structure? So all of these channels are composed from 4 subunits surrounding a centrally located pore. Here we can see a cross-section and 2 subunits. Each subunit contains 6 transmembrane domains divided roughly into 2. The S1 to S4 is the voltage sensor. It's the part of the protein that has lots of at least 4 positive charges that senses membrane potential. And this is connected to the pore domain composed of the S5, pore loop, and S6. So this is the common structure of all voltage-gated potassium or cation channels in the body. However, in the case of KCNH, or in the case of cyclic nucleotide modulated channels, the unique feature resides in the intracellular domain. You can see that there is a C-linker domain followed by a CN BHD, or cyclic nucleotide binding domain. Now, this domain binds cyclic nucleotides, which are a very common second messenger in our cells, and this binding event causes a conformational change that somehow propagates up to the gate, resulting in opening of the channel or modulation of its voltage sensitivity. So this is a common feature of the family. However, in the case of KCNH channels, the ones that cause the long QT syndrome, this domain does not bind cyclic nucleotides. And for many years, it wasn't known why this domain, which shares the same structure as other members of the family, does not bind these nucleotides. And the answer came from the solution of the X-ray structure of this domain, where we showed that it has, in fact, a ligand inside of it. It is occupied by a ligand, and the ligand is part of the protein, or the protein chain, of the polypeptide chain. It's an intrinsic ligand. And no sequence analysis could have predicted that. It's only from structure we were able to understand that these channels are preoccupied with an intrinsic ligand. Just 3 amino acids. Now, another unique feature about the KCNH channels is their amino terminus. The amino terminus contains an EAG domain which is preceded by a pass cap motif. Now this region is crucial for channel function. If we delete it, the channel function is messed up, and so somehow this domain contains information that propagates to the gate, allowing it to open and close in response to voltage stimuli. But how does it do that? So a long time in the field there was a question with what if and with what this domain interacts with. So it could have interacted with the S4, S5 linker, which is the lever that allows the voltage sensor to pull and open the gate, or it could have interacted with the CN BHD, with the cyclic nucleotide binding homology domain here. So during my postdoc, I solved the structure of this domain, but before that, I was able to show that these two domains interact in solution at very low concentrations. For that, I used FSCC, which is a revolutionary method in the field of structural biology, fluorescence detection, size exclusion chromatography. Basically, we load a size exclusion column with any protein we want, and we look only at the fluorescent one, allowing major time saving and high throughputness. And using this method, we were able to show that, in fact, the fluorescently labeled hERG domain is shifting to a lower elution volume when we're mixing it with the cyclic nucleotide binding homology domain, meaning that these two proteins form a complex together. We were able also to measure the affinity of this complex formation using fluorescence anisotropy and came up with this number, 13.2 micromolar, not too low, not too high. And following that, we were able to crystallize something. And these crystals gave a very nice diffraction to around 2 angstrom, but we still weren't sure that the complex is within these crystals. It could have been a crystallization of any one of the components. So we use MALDI time-of-flight, matrix-assisted laser desorption ionization time-of-flight mass spec on these crystals and demonstrated that the two proteins are in fact within the crystal, likely in complex. And they are in complex. This is the structure of the intracellular domain of a KCNH channel. You can see here in green the hERG domain in complex with the blue part, which is the CN BHD, holding in place, the hERG domain holds in place the intrinsic ligand. The hERG domain is what the part of the protein that keeps the intrinsic ligand where it's supposed to be. And yeah. But the question remains, how do these domains, if and how do these domains relate to the non-canonical functions of KCNH channels, and what is non-canonical function? So you can see from our structure, we were able to capture two conformations of this complex within a single crystal. We had a non-crystallographic symmetry, and what you can see here that the interpolation between these two conformations show the flexibility of this intracellular complex. So that means that the intracellular domain of the channel is dynamic, and this complex dynamic may be necessary for the non-canonical signal transduction processes the channel helps with, and that remains to be determined. However, now I'm reaching to the non-canonical functions of these channels. Here you can see the distribution of cancer-associated positions within the intracellular complex of the channels. It's all over the place. So it might be just that you know, cancer cells just accumulate mistakes in the DNA sequence, and that manifests as mutations, but it might be that these mutations change channel function in a way that promotes further cancerogenesis. So why am I saying that? Because when people were expressing hERG channels in cells, they got increased proliferation of any cell types they tried. And this is not because of the fact that these channels are potassium channels or voltage-gated potassium channels, because you see here that in using expression of Shaker, the homotypic voltage-gated potassium channel from Drosophila didn't result in this over-proliferation of cells. So this is something that's unique for KCNH channel. hERG is KCNH, by the way. Another important observation is this one. hERG channels in the normal human body expresses only in the central nervous system, in the brain. However, its RNA and protein is found in every cancer tissue tested to date. So any cancer tissue one biopsies shows expression of hERG channels where they don't belong. And nobody knows why they do that, because lung cells or skin cells are not excitable. They do not change their voltage dramatically, allowing these channels to open or close. So it must be something else. Here you can see possible routes where KV10.1 or KCNH can contribute to cancerogenesis. This is a mystery in the field, how these channels contribute to cellular proliferation, and people are starting to think about using these channels, one as a very early tumor marker, and two as a possible target for anti-cancer drugs. So we are interested in how to inhibit these channels and what's the mechanisms of inhibition of KCNH channels. And one observation we see, we're not the first, but we see that anyway, is that when you co-express hERG channels or KCNH channels with calmodulin, which is a major calcium sensor protein ubiquitous in cells, you get a decrease in the current amplitude. So calmodulin inhibits channel function, and it does that in a calcium-sensitive manner. You can see here that increasing the calcium concentration extracellularly from 1.8 millimolar to 10 millimolar completely inhibits the current that flows through KCNH1 channels. So how does it do that? We invested time in creating a large-scale cancer mutation screening pipeline. We are interested in seeing whether cancer-associated mutations modulate the ability of calmodulin to inhibit the channels. So first, we used yeast to express hERG channels, GFP-labeled hERG channels. You can see here the expression. And then we took these channels and expressed them in another strain that is sensitive to a reduction in the media potassium concentration. So the SGY1528 is a yeast strain that cannot grow without extracellular potassium. It just dies because it lacks the 3 transporters it usually needs to transport potassium into it. However, when we express hERG channels, you can see the recovery of this yeast strain, allowing us to screen now for many mutations simultaneously using deep and following by deep sequencing, and seeing how all of the cancer-associated mutations modulate channel function. And we also want to see how calcium impacts the recovery associated with KCNH, because you can see here that exposure of this yeast to increased concentration of calcium chloride reverses the complementation of the KCNH complementation. Basically, calcium chloride through calmodulin, hopefully inside the yeast, inhibit the channel, which lowers the viability of this strain in low potassium conditions. One more thing we're trying to pursue in the lab is following up on the ability of KCNH inhibition to reduce the proliferation rate of cells. We use a very simple, for now, a proliferation test, and what we see is that while expression of hERG increased the proliferation rate of cells, what I showed you a few slides ago, co-expression with calmodulin reverses or reduces the proliferation rate. So this is the first hint for the effect of calmodulin inhibition on reduced proliferation. We will follow that up with cancer-associated mutations from the e-screen. Now, how does calmodulin, calcium calmodulin, inhibit the channel? We are very interested in understanding the exact molecular mechanism by which binding of calmodulin to the intracellular domain of the channel result in obstruction of the gate, result in lack of channel activity. So we do that by a variety of molecular approaches, but first, 3 binding sites for calcium calmodulin were recently identified in the amino and C termini of the channels, the BDN1 in the amino terminus and the BDC1C2 and the recently we discovered another site at the PAS-CAP domain. So we will concentrate in the next few slides on the BDN1 and try to understand how calcium calmodulin inhibit these channels and whether cancer-associated mutations within this domain alter the interaction with, of the channel with calcium calmodulin. So this is the part of the channel we are pursuing at the moment. We were able to express it. We were able to generate a complex, a calcium-dependent complex of this domain along with calcium calmodulin. We increased the production scale and got buckets of protein. And then we were able, using, again, fluorescence, this time of Dansil, a fluorescently labeled calmodulin molecule, to demonstrate that the affinity of calmodulin to the BDN1 is very high, about 467 nanomolar, and we have a stable complex in solution. We show that using FSCC here, and here we do some competition with unlabeled calmodulin to verify this KD we have. And using ITC, another biophysical method that is independent of any fluorescent labeling or labeling whatsoever, we were able to show a complex formation between calcium calmodulin and a short and a long versions of the N-terminus with very comparable affinities. We did that. We show the calcium dependence of this complex formation using analytical ultracentrifugation. Here you can see that without calcium, in the presence of 10 millimolar EGTA, there is no complex formation. However, when we increase the calcium concentration, we have an increase in the population with the mass equivalent for a complex between calcium calmodulin and the N-terminus. This is some SEC mouse results showing the same thing. And finally, we did a small angle X-ray scattering in the ESRF, demonstrating the structure of the complex, the low resolution structure of the complex between the amino terminus of the channel and calcium calmodulin. You can see the molecular envelope sits very well with this molecular model. And however, interestingly, this is the complex between the C-terminus and the N-terminus of the channel, the one I showed you the X-ray structure of, and it doesn't sit very well with the envelope in solution, indicating that in solution, unlike in the crystal, this complex adopts a more elongated conformation, which is still mysterious, but might be important for channel function. But doing all that, we did this fiddling here, and the huge piano fell from the sky in the form of this thing. This is the Cryo-EM structure of an intact KCNH channel in complex with comodulin. It came from the McKinnon lab a few months ago. McKinnon is the Nobel Prize laureate for the initial structure of a potassium channel. And what this structure shows, amazingly, is how comodulin interacts, basically grab together the amino terminus and the carboxy terminus of this channel. However, this part of the structure, I remind you this is a cryo-EM, is in 6 angstroms. So we don't have still the molecular determinants necessary for this interaction. It's all speculation around here. However, if you look at this structure closely, you can see here the cancer-associated mutations that are scattered around the amino terminus and might interfere, some of them, with the binding of calcium comodulin. And we set up to test that, and especially two mutations that are in very close proximity to comodulin, the G147V and the A169D. And here, using electrophysiology, which is another expertise of the lab, we recorded the currents that are flowing through these channels in response to application of ionomycin, which is a calcium, a synthetic calcium channel that allows calcium to specifically go into the cells. So you can see here in the wild type, after application of calcium, there is an inhibition of the current, which is somehow recovers after a while. We are still not sure about this part. It might be related to additional modulation of the channel. However, the G147V, a cancer-associated mutation, results in decreased inhibition of decreased calcium-dependent inhibition of the current. And this is true for 169 as well. However, 158, which is not in a direct contact or in a close proximity to comodulin and lobe, according to the cryo-EM structure, does not do anything to the calcium-dependent inhibition. And this is a summary of the data. All of these mutations do not change, almost do not change the biophysical properties of the channels. They do not change the voltage dependence according to this conductance voltage diagram. However, so the answer is yes to do cancer mutations modify channel inhibition by calcium for at least some positions in close proximity to comodulin. The answer is yes, they do modify that. But we are on the way conducting cellular validation assays of these mutations. One last thing, I have 1 minute. So what is the mechanism? Why calcium comodulin, how calcium comodulin inhibit the channel? This is still unknown. How the binding of this protein to the intracellular domain modify the gating of the channel. And recently, in the summer, a couple of papers came out demonstrating the molecular cause of this horrible syndrome. This is the Zimmerman-Laban syndrome, is a very severe developmental syndrome of kids, as you can see with these abnormalities, and a very severe epilepsy. They die very young. And these papers that came out are the first papers that showed the contribution of a KCNH channel to CNS function. There is, although these channels are expressed in the brain, there is no known function for this channel there. And here they showed that this point mutation caused a gain of function of the channel, meaning that the channel is more easily opened, more conductive. And this point mutation results in this horrible syndrome. And what we show using these mutations that were found is that here you can see the current that is generated by this point mutation of zero-molar band. This channel is basically open all the time. It's not voltage sensitive, almost not voltage sensitive. However, after application of calcium, it retains its voltage sensitivity. It has a major shift in the GV curve. So it has kinetics again, and it opens almost like in the voltage span of wild type. And what we think, we are in the process of proving it, that in the case of wild type channels, what calmodulin is doing is producing a major shift in the GV, in the voltage dependence of the channel to regions so depolarized that are beyond where cells are can reach, and leaving the channels in a closed state. So this might be a mechanism that allows calcium calmodulin to inhibit the channel, this shift. And by that, I will going to finish and thank some of the lab members that help contribute to this ongoing project and these funding agencies. Thank you.
Thank you. Open for questions.
Lunchtime.
Why have some hearts and some broken hearts?
Because Shachar found a job, and Monica was an intern from Heidelberg, a very gifted intern for 6 months, and went back to Germany. And Moshe Giladi is always here, but it's not always good.
Any question? So I have a question I have to ask. You show that these ion channels were highly expressed in the brains, and you show that they have mutations all over in different tumors, but what about gliomas or brain tumors? Or?
Yes, they're definitely expressed in those tumors. However, obviously, it's a little hard to separate a pattern of expression from a tumor cell that resides within the hosting tissue. So I don't think anyone looked at specifically if there is an increase in expression in gliomas compared to normal brain, but my hunch is that it must be.
Yeah, I was looking online in the CBio portal, TCGA, and I saw there are mutations, specific missense mutations, specifically in glioma for the channel. Any other question? Okay, so I leave. Thank you very much for a great session.
Okay, so I'd like to conclude the event. It was very exciting. Just some numbers. We had more than 20 lectures, 7 sessions, 7 meals, which is very important. And who counts the calories? How much calories? You don't want to know. Anyway, I hope you enjoyed the conference and you got some update about what your friends in the university are doing. And I hope you had some time to enjoy the weather, which is not less important. None of this could happen without Mickey's help. Which did great work in management of this entire conference and management of us, and Vered will say several more words.
No, just thank you, Mickey. It's been amazing, and she made this conference really happen. We just did the scientific part. She did everything else. And I would also like to thank Ruti from Ortal, which helped organize everything. And thank you all.